So good afternoon, everyone. My name is Victoire Viannay. I'm CEO at Fluicell AB. And this is Gavin Jeffries, he's CTO and founder. And we are going to present you Fluicell. So Fluicell AB is a biotech company from Göteborg, founded in 2012 as a spin-off from Chalmers University of Technology. We are specialized in developing microtechnology products for the life science market. We are a publicly traded company listed on Nasdaq First North since April 2018. So Fluicell is employing 20 employees. Our board of directors is comprised of 5 members who have expertise within business, law, and science. Our chairman of the board is called Stefan Tilk, he's CEO at Neves. Since July 2019, a scientific advisory board has been appointed. It's comprised of 3 medical doctors who have expertise within transplantation surgery, dermatology, surgery, and drug development. Their advice is extremely important for our company because it's going to help us with the future development of the company. So why is there a paradigm shift in biomedical research? So pharma companies want to deliver high patient value solutions, tailored treatment strategies. For that, they need for faster, more relevant information sooner in the discovery process and a better utilization of high-value samples. And that is exactly what we can provide at Fluicell. Our product generates unique and critical data from individual cells early in the drug discovery process. So our core expertise is called microfluidics. So microfluidics can be explained as the capacity to trick a small amount of liquid so it acts like a solid. And we are using this physics advanced phenomenon to create an easy-to-use product with a lot of application of utility for the life science. So on this slide, you can see our current product portfolio. So first of all, the biopen on which the company was built upon. This is a single-cell biology tool. It means that it can interrogate individual cells or a small group of cells. The second product is called Dynaflow Resolve. It's not a product that we have been developing ourselves. It's a secondary drug screening platform, and we have the right to sell it through a worldwide and exclusive distributor license agreement. Finally, the Pixlr, it's our newly launched product. It's a bioprinting platform. It's a 3D single-cell bioprinter. So on this slide, you can see that we are addressing a market globally, but most of our customers are currently in the USA and in Europe, with few exceptions in Asia and in Australia. So we have very reputable clients within university, research institute, and pharma companies who like and use our product. And this key opinion leader has been giving us some testimonial to show us that they like our product, which is extremely important for us because it shows that our products are reliable and it will certainly gather followers. So as well as scientific publication, so this is only a sample of scientific publication. This is extremely important also for the company because researchers around the world in academia, but also in pharma, are reading the scientific publication and make buying decisions upon that. So we believe that having a very strong IP portfolio is crucial for a company like ours. So we have a patent portfolio comprised of 6 patent families with currently 14 approved patents. There's a lot of patent applications still ongoing. The patents are protecting both the technology but also the method. And it's extremely important to both protect the current product but also the future product to come. And it gives a great value to the company.
Okay, thank you, Victoire. So I'm going to explain a little bit more about what Victoire was saying with regards to our product portfolio. So the BioPen system, which was our first product, essentially, it's what took the company public, is a single-cell liquid delivery tool. What this means is that it allows you to deliver compounds to one or a few cells in a very, very controlled way. By doing this in this very localized and controlled way, you can really decide on how much a compound or drug each cell gets. And you can do this in a way where there's no contamination to any other part of the dish. And this is something which is used a lot within toxicology and things. And it's equally why we're picked up and used in the FDA and the NIH and things like this. The BioPen, being a single-cell delivery tool, has a broad application space. And by that, we see users within these various areas of oncology, enzymology. We get quite a lot of interest from electrophysiology as well, where people are actually trying to understand how the modes of action of drugs. And this kind of broad application space we see kind of expanding as we go forward. Now we're starting to see some ex vivo implementations of the device. Here is one of our kind of more latest ones where they're actually studying the effects of blood and things on mosquito needles with the goal of trying to understand how to control how mosquitoes feed from human blood. And this was done with a very well-known person from actually Rockefeller University who's really focusing on the malaria and Zika. Our second one that Victoire kind of introduced is the Dynaflow Resolve system. And this is seen by many as the quintessential secondary ion screening platform. This is something that we took over the complete license of, and actually it became a complementary technology within our portfolio, allowing us to kind of integrate a little bit more and a little bit more expansively with the not only the academic, but also the commercial kind of workflows of the pharma industry. With this, we essentially have a dynamic and a very robust microfluidic platform, and this platform allows you to understand and study any type of cell in any kind of typical patch clamp configuration. And with this, you're able to get very, very profound data. And we're very happy to actually see, and Victoire equally touched on a little bit, to actually see that communication within this field is very important. And actually to see publications coming out from some of our kind of core leaders. So this is actually from Genentech that was recently published in Science. And their whole studies are based on the Dynaflow platform because they can get this kind of unprecedented data. So this being said, what a lot of people are actually interested to hear about is BioPixlr. Now, BioPixlr is something which is completely new. It's completely developed in-house by Fluicell. And we see this as essentially our kind of vision of high-resolution bioprinting for biological tissue generation. This was developed as an all-in-one discovery platform. This doesn't require any other parts or pieces from anywhere. This allows you to essentially print tissues all in one place. And it does this in a manner which we utilize microfluidics. So microfluidics is our core expertise, and BioPixlr stands alone in its ability to print individual cells, to build these biological tissues. And what we can actually see a little bit more in depth here is that not only are we depositing these individual cells, but we're doing it without any gel. So this we see as a new concept of bioprinting. We're building tissues, we're building these structures, we're doing it with a microfluidic structure. This microfluidics is a consumable, it's a disposable, meaning we can stay within standard GLP systems, and it gives us the ability to create detailed, multicellular, and complex biological tissues, some of which I'll kind of get into in just a second. So just to give a little idea of what we're doing here, this is a very simple approach. So this is just demonstrating a very small area of printed cells. So we can see the print ahead here. And one thing that you can see in connection to other bioprinters is we can actually see all of the cells being printed in real time. We can switch between different cell types. All of the cells are actually housed within the printhead, just like they would be in an inkjet printer at home. We can then print and deposit these cells in any particular orientation or position that we would like. And in a second, it'll show you right at the end that these actually have a particular fluorescent label in them. So we can actually see these differences. So now it should go, there we go. So we can actually see the differences between the cells and how they were printed. The BioPixlr approach essentially is to print the microenvironment that the cells are going to grow into. It's very important for complex tissue generation and complex biological model generation to actually put the cells where you want to have them so that you can have control over the complete environment. We do this in a way where we can actually follow the building of these biological tissues because we don't have anything which essentially limits the cell-to-cell interaction. Because we utilize microfluidics, we can do this, as it says at the bottom, with very small volume usage, very small numbers of cells, and as we're doing it in a microfluidic, gel-free manner, we have very minimal handling of the cells. So we can print small things or large things. Here's just a couple of examples that have come out recently. On the left side is essentially the structure that was printed. On the right side, essentially, is the grown models. So most of the models that we work with and that we work with people on require very fast turnaround. These are what would be called first approximation models of these cancers or in fact, of these tissues. However, we can see quite easily from these that within 24 hours, we're starting to get fully integrated tissue models. This can be done in a very simplistic way, like we just saw, or we can, in fact, go to very large, very more complex ones, where we actually start to mimic histology examples. So we can actually, cell by cell, as you can see, all these little dots are individual cells. We can change the cell types, and we can print just the same way that you would see on a kind of conventional printer. But rather than using any sort of ink on paper, we're actually using biological or human cells on top of a surface. So we can mimic tissue examples, we can mimic simple model systems, but I mean the core element is that we're able to generate these both simplistic and complex structures with very small numbers of cells. We fully utilize our expertise of microfluidics so that we can handle not only the cells, but also the liquid around it. And we do this in a completely gel-free environment so that we can control the microenvironment in which the cells are going to grow. And this is very important if you need that very high precision and high fidelity printing, which is quite often needed when you have very small numbers of cells or you want to start having very individualized models.
So I will start now to talk again about the business development. So Fluicell is targeting two market segments, the pharma, pre-medical research, and the academic research. So biopharma industry is facing a lot of challenges, and we can address some of these challenges with our product by improving decision early in the drug discovery process. But we're also targeting the academy. Academia is a field which is global with very strong financial support. The sales cycles are longer than in the pharma industry. However, the academic is attracting both private and public funding. For example, Horizon in the EU or NIH in the USA. Also, something important also regarding academia is that they are keen on testing new methods and technologies, so they are early adopter. And as we were explaining, they are publishing scientific paper, which is extremely important to gather followers. So all the market analysis outline that the most accessible market today for the bioprinting company is actually the academic market, which represents more than 80%. So as Gavin was explaining, the bioprinting platform BioPixlr is a quite new and unique way of doing bioprinting, and it's clearly addressing an unmet need within bioprinting. So single cell resolution and maximal usage of patient-derived sample is an important requirement for building pre-medical models. And these models are expected to play a critical role for drug discovery application in the future. So BioPixlr, it's one product, but it's several business opportunities within drug development, as we were saying, but also regenerative medicine, and in the future, transplantation science and biointegration. So that's the competitive landscape. For the BioPen and the Dynaflow, there is no direct competitors. Those are sole source products. Regarding the BioPixlr, it's positioned in an emerging, yet quite competitive market. However, most of the competitive technology are located within bioextrusion and inkjet. The BioPixlr is in a different, yet highly relevant category. And the market is moving towards high-precision printing, which is our direct target. So there is a lot of competitive advantages to the BioPixlr. So as Gavin was saying, single-cell resolution, high precision, high reproducibility. And also, with the BioPixlr, more than 95% of the cells are surviving. It is extremely important because the cells are completely alive after the printing process. It is very good for small and rare sample utilization, and we are building multicellular tissue models. So this is our revenue models. So the three products, BioPen, Dynaflow, and BioPixlr, are sold at platform. This is a one-off revenue. Then, in addition, we have recurrent revenue from the sale of the consumables. So the BioPen tips and the BioPixlr tips are a one-time use consumable, whereas the Dynaflow chip is leasable. Each leasing agreement lasts up to 4 years and then can be renewed. In addition, we are offering service, we are looking for partnership, and we have been starting to do contract research organization type of work with the BioPixlr. So the market positioning within bioprinted is divided into 3 segments. So the market has been dominated so far by low-cost desktop bioprinter, which is with simple feature. It represents typically a bioprinter which has price at less than $10,000 US. However, due to the need for automation, the market is not switching from the first segment toward the last segment, where you have the more industrial bioprinter. So BioPixlr is a printer which is offering features which only are available in the third category. It's an all-in-one platform, as Gavin was explaining. So by positioning the BioPixlr within the second segment, we believe we have a very competitive product. The growth potential for our current product is good. We are targeting a market which is expanding well and is with a double-digit CAGR, sorry. The BioPixlr is addressing a mass market. Conservatively, it worth $1.3 billion US, and also it's growing at a double-digit CAGR. For the future, we have a disruptive core technology, which means that we can develop more product, of course, but our vision is to target regenerative medicine. For regenerative medicine, the total addressable market in 2024 will be around $40 billion US. So our sales strategy. So we have been completely remodeling our sales strategy. Before, we were going through a full distributor model, and we realized that it was not working very well for us. So we have been renegotiating all the distributor agreements from being exclusive to non-exclusive agreement, apart for Israel, which is still exclusive. By doing that, we could retake our hand on our market because we are the one who have the most knowledge in how selling our product. So we have been increasing our sales team, and we have been increasing our global presence and lead acquisition through direct contact, conferences, and digital marketing. And the sale process has been adapted to each product and customer segment. So we start to see some positive response to our new sales strategy, which was put in place around a year ago. And we really believe that next year we are going to have a much more aggressive growth. Thank you.
Tack så mycket. Spännande.
Tack.
Imponerande. Tack. Komplicerat.
Complicated? Ja, det är lite komplicerat.
Complicated.
Ja. Har vi någon fråga här? Ja, det har vi. Det har vi.
I just want to know where you stand in this Pixlr, this regenerative medicine. One thing is to put a number of layers of cell on top of each other, but in order to get a functioning tissue, you need intercellular matrix like a scaffolding. Yep. Otherwise, in my view, nothing much is won. What can you do in that respect?
You're absolutely right. I mean, within regenerative areas, you need to get a significantly more complex infrastructure in it. So most of what we were showing here is largely 2D work, and some of it is essentially 3D, but we have the same ability to print in 3 dimensions as we do in 2 dimensions. So we have a particular type of extracellular matrix and cell binding system that will allow us to essentially build layer by layer structures so that we can actually add layers of complexity. One thing which